Okay, this slide has to do with a question I had asked in the last video, but the question being, why are there so many stinking steps in these things? Why can't it just be a molecule enters a cell and activates what it's supposed to activate? Like, why does it have to be a phosphorylation cascade and all these intermediates and activating things and then deactivating them and all of that? And there's a couple benefits to it. So when you ask, like, what's the reasoning behind something happening, that gets kind of hairy in biology. But anyway, if you say, what are the benefits of? So one of the benefits that scientists find, I'm going to slide myself over here, one of the benefits that scientists find in having a lot of steps, one is it allows the signal to be amplified, they call it. And anytime you amplify something, it just means you're making it more impactful. If you amplify sound, you take a small sound, and when you amplify it, it becomes really loud. Well, in this case, we can take this goes back to the epinephrine one over here. They have written down numbers of molecules, quantity of molecules. And if I look at the epinephrine, one molecule can hit a cell, just one molecule, and it's going to activate a G protein, but it can actually activate several hundred G proteins just from that one molecule landing on the receptor. So you get to 10 to the second molecules, which is about, it's several hundred molecules being activated of the G protein. Each of those G proteins can then go and activate the adenylyl cyclase. Okay, so we're at several hundred of those. But then when you start the adenylyl cyclase that's activated, it can take a lot of ATP and turn it into cyclic AMP. And so when we do that, all of a sudden we get to 10 to the fourth, which gets to tens of thousands of molecules of cyclic AMP, which are going to then activate tens of thousands of molecules of protein kinase A. But then that protein kinase A can activate several molecules of the next protein kinase. So then you get 10 to the fifth, you're getting to hundreds of thousands of molecules that are activated, go to the next protein kinase, or the next step here, the glycogen phosphorylase, and you get up into the millions of glycogen phosphorylases that are activated, which means we had one epinephrine molecule land on this cell, and we're going to end up having millions of glycogen molecules turned into glucose 1-phosphate, and that can happen a lot of times. You get to 10 to the 8th, that's 100 million molecules of glucose 1-phosphate being made from this one epinephrine. So this becomes pretty powerful, this amplification idea. And so that's one of the advantages of having a bunch of steps, is that instead of having one molecule making one molecule of epinephrine resulting in one glucose 1-phosphate being produced, it's ending up getting hundreds of millions of the product that we want. Okay, so that's a really good thing, really powerful thing. So there's that amplification idea. The next idea here is that by having a lot of steps, it actually increases the diversity of responses that you could get from one signal, and by having a diversity of ways that it could be interpreted, it ends up meaning that any cell, specific cells within the body, can all have their own specific response to a signal. So what this is showing is 4 different responses to the same signal. So actually the first and second and third, they're all using the same receptor protein, but then we have the fourth situation over here, 1, 2, 3, 4, response 5, is a different receptor protein. Okay, and so there's a cell that would have a specific situation there. We can also look at, if I compare 1 and 2, and these would be different cells, but they've got the same initial receptor, and they're just resulting in different sets of molecules going down through them. Anyway, I'm not going to go into the detail on all of this, but it gives us more options of how a single protein or a single molecule can be interpreted by cells. And that's how you get something like the liver cells respond to epinephrine by making sugar available to the blood, but the heart cells increase how often they pulse. Okay, so your heart rate increases and becomes more powerful actually when epinephrine is released. So you have 2 different responses because we have all these steps. If it was something as simple as we've got a signal molecule, it enters a cell and it does its one thing, there'd be a limited number of responses we could have for the one molecule that it's talking about. So that's some of the answers to why there's so many steps. It just gives us some advantages along the way.